Next, the approach to target identification, and at last, we are gonna look at tools for target identification and validation. Let me start this seminar by saying that identifying the biological origin of a disease and the potential target for intervention is the first step for the discovery of medicine. Target identification is the process of identifying the direct molecular target, for example, protein or nucleic acid of a small molecule. In clinical pharmacology, target identification is aimed at finding the accuracy target of a drug. There is need to find a protein, for example, the receptor or gene associated with the disease with which a potential drug interacts, so-called the target. Not every target is equally capable of affecting the course of a disease. Now let's take a look at the steps in drug discovery or design. The drug discovery process starts with the identification of a molecular target, and next is the target validation. During target validation, it associates with the specific disease and is its ability to regulate biological processes in the test body. The target validation confirms that the interactions with the target produces the desired change in the behavior of the disease cells. It is a crucial step in drug discovery process. Identification of new drug targets, target validation, bioprocess assay developed followed by lead identification provides very important input in the development of new potential drug candidates. Now let's see about the characteristics of a good drug target. The drug target is a biological molecule, normally a protein that could exist in isolated or complex modality. The biomolecules have special sites that match other molecules. These molecules could be endogenous substances such as chemical molecules or drugs. The biomolecule structure might change when the biomolecule binds to small molecules, and the change in structure normally are irreversible. Following the change in the biomolecular structure, various physiological responses occur and induce regulation of the cell, organ, tissue, or body status. The physiological responses triggered by these changes in biomolecule structures play a major role in complex regulation and have a therapeutic effect on the pathological conditions. The expression's activity and the structure of biomolecule might change over the duration of pathological processes. Small molecules binding to the biomolecules are called the drugs. Now let's see about the differences in traditional drug designing versus the new strategies in drug designing. The path to identifying and validating the drug candidate molecule is not a one-size-fits-all diseases strategy. The first stage in drug discovery process is to understand the disease mechanism using cellular and genetic approaches in order to identify potential drug targets. Now let's see the role of target identification in drug discovery. Target identification and mechanism of action studies play an important role in the small molecule discovery. A good target needs to be safe, meet clinical and commercial needs, and it should be druggable. A druggable target is accessible to the putative drug molecule, be that a small molecule or large biological molecule, and upon binding a biological response which may be measured both in vitro and in vivo. Good target identification and validation enables increased confidence in the relationship between the target and the disease and allows us to explore whether target modulation will lead to the mechanism-based side effect. Current therapy is based on less than 500 molecular targets of above 10,000 possible targets, 45% of which are G-protein coupled receptors, 28% are enzymes, 11% are hormones and factors, 5% are ion channels, and 2% are nuclear receptors. Target identification can be approached by direct biological method, genetic interactions, or computational interference. Combinations these approaches may be required to fully characterize mechanism of small molecular action. According to Board Institute, using a multi-phased approach to the target identification problem in the context of genome-based drug discovery include quantitative proteomics based on mass spectrometry, genetic complementation of small molecules, effect using RNA interference, combinational interference by connective analysis using reference compounds. A forward genetic approach is characterized by identification, often under experimental section pressure, for a prototype of interest, followed by identification of the gene responsible for the prototype. Now let's see the approaches to target identifications. There are three distinct and complementary approaches for discovering the protein target of a small molecules. The first one is the direct biological method. Direct method involves labeling the protein or small molecule of interest, incubating of the two population, direct detection of binding, usually following some type of washing procedure. Genetic interaction methods. Genetic manipulation can also be used to identify protein targets by modulating presumed targets in cells, thereby changing small molecular sensitivity. Comparative genomic strategy aim to compare simultaneously two or more genomes in order to identify similarities and differences and hence identify potential drug targets. Computational interference method. Target hypothesis, in contrast, can be generated by computational interference methods. Target hypothesis, in contrast, we can be generated by computational interference, using pattern recognitions to compare small molecular effects to those of the known reference molecule. Now let's see the tools for target identification and validation. Reliable technologies for addressing target identification and validations are the foundation of successful drug development. Microarray has been well utilized in genomics or proteomics approaches for gene or protein expression proliferating and tissue or cell scale target validation. Antisense technology, that is the RNA interference, enables sequence-based gene knockout at the RNA level. Zinc finger proteins are a DNA transcription targeted version of knockdown. Chemical genomics and proteomics are emerging tools for generating phenotype changes, thus leading to target and hit identification. Target identifications with proteomics is performed by comparing the protein expression level in normal and diseased tissues. NMR-based screening as well as activity-based protein profiling are trying to meet the requirements of a high-throughput target identification. Now, one by one, let's see the tools for target identification and validation. The first one is the microarray. Target identification seeks to identify new target, normally protein or DNA or RNA, whose modulation might inhibit or reverse disease progressions. Current technologies enable researchers to attend the correlation changes in genomic and proteomic expression with human disease in hope to finding a new target. Assess gene and protein expression to identify novel targets and can also be used to validate the found targets at the tissue and cell scale. In microarrays, nucleic acid microarray, which primarily uses short oligonucleotides, long oligonucleotides, and PCR amplified cDNAs as array elements, are their dominant because of their relative easy synthesis and chemical robustness of DNA. Data generated from genome sequencing project is in several organisms have provided the opportunity to build comprehensive maps of transcriptional regulation. Array-based gene expression analysis has enabled parallel monitoring of cell transcription at the level of the genome. Thus, nucleic acid microarrays have a significant impact on our understanding of normal and abnormal cell biochemistry and thus on the choice of target for drug design. Next, let's see about protein microarrays. Because most drug targets are proteins, protein and peptide microarrays are said to have an important impact on the drug discovery. Protein arrays and emerging yet very promising technologies are now used to examine uh, enzyme substrate, DNA, protein, and protein interactions by profiling the differential expressions of protein using antibody arrays and correlating the changes to a disease prototype. Putative targets to a particular disease can be identified. Such microarrays have not yet been used to their fullest potential because of the difficulties in the technology. Now let's talk about tissue and cell microarrays. An alternate to the use of whole tissue specimen is the use of live cell microarrays, which can be used to identify potential drug targets by functioning characterizing large amount or large number of gene products in cell-based assays. Next is the antisense technology. A key strategy in target validation is to determine what happens with respect to the phenotype or the expression of the other genes in cell or model organism. If a gene of interest is either deleted or its activity is inhibited. Gene knockout mimics the activity of drug that completely inhibits the normal function of the gene product. Antisense oligonucleotides. Complementary to a portion of target mRNA molecules, oligonucleotides are the original type of molecules used for blocking protein synthesis of the target mRNA and thus achieving the knockdown of the target gene. One example is the identification of COX-17 as a therapeutic target for non-small cell lung cells. Now let's see the zinc finger proteins. Zinc finger proteins have remarkable versatility for recognizing different sequences of DNA and variations in the amino acid sequence of the C2H2 domains allow them to be targeted to different locations in the genome. Each zinc finger is a short stretch of 30 amino acids containing 2 conserved cysteines and 2 conserved histidines. These proteins have been used as the DNA binding domains for novel transcriptional factors. Zinc finger protein transcriptional factors can be applied to potential new drug target validation in two ways. One way is the direct way involving designing zinc finger proteins to validate the phenotype effect of activating or repressing a gene. Alternatively, libraries of zinc finger protein transcription factor might be used to screen cells of desired phenotype changes. In chemical genomics and proteomics, rather than finding drugs for target in the conventional pharmacological approach, forward chemical genomics in a sense finds target for known drugs. Its goal is to discover the specific molecular targets and pathway that are moderated by particular chemical molecules. That is, study the biochemistry underlying the phenotype changes included by the chemicals. That is to study the biochemistry underlying the phenotype changes included by chemicals. Now let's see about target library approach. Various kind of chemicals have been used to generate novel chemotypes and once an effect is found, the next step is to identify the biological target using a affinity matrix made up of immobilized hit compounds. High-throughput NMR-based screening is also utilized for fast identification of drug-target interactions. Thank you.